Hello everyone, my name is Alex. I'm the head of project management team for antibody drug discovery at GenScript ProBio. It's my pleasure to discuss our antibody engineering and developability services in today's session. Good functionality and developability are critical characteristics for therapeutic antibodies. A therapeutic antibody should not only have good binding affinity, specificity, in vitro and in vivo potency, but also should good chemical and physical stability and developabilities. They are very important in the downstream drug development and manufacture. GenScript ProBio has extensive capability in structure-based modeling design for meet-better leads. We have access to various database and MOE platform. In the aspect of developability, we provide antibody humanization, developability assessment, and optimization services. Regarding functionality, we provide Affinity Maturation and FC engineering services. Next, I will cover our primary biologics engineering services, starting from humanization. Our humanization services use traditional CDR grafting and back mutation strategies. There are two featured service packages in ProBio. One is Express package, which takes as fast as 2.5 weeks to get your humanized antibodies. The other is Deluxe package, which takes about 8 weeks to get humanized antibodies with no affinity loss and potentially lower immunogenicity. Here is the workflow for Express humanization service. We start with humanization design and PTM analysis. The humanization design could be finished in about 2 days. Then we will initiate chimeric and humanized antibody production and purification, which takes about 2 weeks. Around 9 humanized variants will be produced, then tested in BRCO8K for affinity analysis, which takes about another 1 to 2 days. We are the #1 fastest service provider in the world with comprehensive developability prediction report, and it's guaranteed that at least one humanized variant show comparable affinity compared with chimeric antibody. For the Deluxe package, we will construct a mutation libraries in FRCIPA system for humanized variants screening and affinity ranking. FRCIPA is a patent high-throughput screening platform to express soluble antibodies in the meantime, screening affinity, thermal stability, expression level, binding epitopes with no royalty burden. Due to the larger screening pool in comparison to Express package, we can ensure no loss of affinity for the humanized variants. Additionally, we have the potential to identify candidates with minimal back mutation sites, and it potentially resulting in lower immunogenicity. Here is a summary of the two humanization packages. The Express one takes about 2 to 3 weeks, and the Deluxe one takes about 8 to 11 weeks. The deliverables include sequence of the all humanized variants and 0.2 mix of top 3 humanized antibodies. The developability report, including PDM analysis, immunogenicity prediction, and aggregation analysis, is optional. We have successfully delivered over 500 humanization services, and here are the 4 case studies. In the first case study is a mouse IgG humanization with BRCO determination. The humanized variants show comparable affinity as the parental antibody. In the second case study, FACS test was performed to evaluate the binding between the humanized and the chimeric antibodies. and the same binding profile was observed based on the FACS results. The case study 3 is a humanization for single-domain antibodies. The affinity is maximally retained by rational design. And in the case study 4, is a humanization is actually a canonizations for a mouse antibody. In summary, we have 2 humanized service packages and are able to get humanized antibodies as fast as 2.5 weeks. And we have successfully delivered over 500 projects. The most advanced project is marketed. Next, we will discuss our affinity maturation services. The success criteria for affinity tuning is to reach the desired affinity with minimal mutagenesis. And the key supporting platforms including high-quality mutagenesis libraries and powerful screening platforms. The mainstream library construction approach are summarized in the table here. NK libraries is using single nucleotide synthesis primer, which leads to redundant mutagenesis and stop codons, resulting in truncated products and poor amino acid distribution uniformity. There is high likelihood of losing high-quality mutants during the later screening stage, while the trimer libraries is introducing a mixture of all 20 amino acids and the mutant position, which avoids the issues such as codon bias, frame shift mutagenesis, and control of stop codon incorporation. The approach enhances the library homogeneity and coverage, leading to a higher precision and accuracy in substance screening. The precise mutagenesis library is utilizing semiconductor-based oligosynthesis technology. PML provides precise control over every synthesized variant, resulting in a diverse, comprehensive mutagenesis libraries with unbiased distribution. This saves variable screening time, accelerates the discovery workflow, and increases the screening success rate. Overall, the trimer and the PML library are highly recommended for constructing libraries for affinity maturation. Surgical ProBio provides 2 featured service packages for affinity maturation, utilizing the PML and the trimer platforms respectively. The deluxe package includes the MOE-based structural modeling, precise site saturation mutagenesis library construction, combinational mutagenesis library construction, and FACS screening. The overall timeline is about 14 to 16 weeks, with more than a tenfold improvement in affinity in most cases. While the standard service package involves MOE-based structural modeling, trimer combinatorial mutagenesis libraries, and planning and screening by phage display platform. The overall timeline for the standard package is about 12 to 14 weeks, and it's ensured that a more than 5-fold improvement. We have an integrated computer-aided molecular design platform and extensive experience to guide us for identifying the key residues and the mutated amino acids for the library construction. The MOE software directly connects to the database such as RCSB and PDB, and also includes over a dozen classical protein family structures database, which is enabling us to do efficient structure simulation. The deluxe package, as we mentioned, is based on the precise site saturation mutagenesis library and the combinatorial mutagenesis libraries and the FACS platform. which provides a comprehensive screening strategies for affinity maturation. After identifying the key residues from the MOE modeling design, the PML libraries will be constructed for each of the key residues in the CDR region. And then the PML is a semiconductor-based oligosynthesis technology, which ensures the even distributions of 18 amino acids in each of the PML libraries without stop codon or unexpected codon. FASIPA is a high-throughput screening platform introduced from national research consoles in Canada. There is a SASA tag contained in the construct, which is anti-BRCA single-domain antibody sequence with picomolar affinity against BRCA. Thus, we can utilize the interaction between BRCA and the FASIPA supernatant for high-throughput screening. For example, in BRCA system, the BRCA chip could be used to enrich the FASIPA supernatant. Then the antigen could flow over the chip for off-rate ranking. The FASIPA system greatly reduced the cost of recombinant expression and ensure the success rate of the screening. For the standard service package, it utilized the trimer library and field display platform. The trimer is created by linking 3 nucleosides, where each trimer represents a single amino acid. By designing the trimer mixture, the library simultaneously mutates multiple amino acid sites in the target region to produce different mutagenesis combinations. The features of trimer libraries include adaptive codon optimization, no unexpected codons or stop codons, highly uniform distribution, and customized amino acid mutagenesis type and proportions. Here is one case study with trimer combinatorial mutagenesis library. The affinity is improved from 10 to -8 to 10 to -10 with a 165-fold improvement. The 3 case studies here are using PML libraries plus FASIPA screening strategies. In the case study 2, the affinity was improved from 10 to -10 to 10 to -7 to 10 to -10. In the case study 3, the affinity was improved from 10 to -10 to 10 to -12. And case study 4 is a single-domain antibodies. The affinity was improved from 10 to -7 to 10 to -8. Besides human IgG and single-domain antibody, we also have experience on affinity maturation for other species antibodies, including feline, canine, and rabbits. Here are 3 case studies in which the antibody affinity has been improved about 20 to 200-fold. In summary, ProBio has over 110 project experience on affinity maturation. The deluxe package includes the precise site saturation mutagenesis library, so-called PML, plus the combinatory mutagenesis libraries and the FASIPA system screening. It's guaranteed a 10-fold affinity improvement for most cases, while the standard package includes the trimer combinatorial mutagenesis library and the field display platform, and it's guaranteed at least 5-fold improvement. Next, let's briefly talk about FC engineering. In general, FC engineering includes reducing or enhancing effector functions and half-life extension. For reducing the effector function, ProBio is partnered with MapSoft to offer STR-silenced antibodies. STR-silence is a technology to truly abolish the FC domain effector function. It's almost up to 100% silence. Compared with other FC silence, technologies such as LALA or glycosylation, which only reduce but not truly 100% abolish the Fc effector binding. For enhancing effector function, Transcript ProBio provides a variety of Fc engineering solutions for ADCC, CDC, and ADCP enhancement. Regarding half-life extension, we can either use the FTO mutants or conduct library screening based on the project needs. Here are some data from the STR mutation. From both binding assays and cell-based assays, the STR shows the least activities against Fc gamma receptors while retain the binding to FcRn. Last, we'll discuss our durability assessment and optimization services. We have 3 packages for durability assessment, including in silico durability analysis, in vitro durability assays, and the manufacturability assessment. The in silico durability analysis includes immunogenicity prediction, post-translational modification analysis, and aggregation analysis, which takes about 1 week and generally be done after obtaining the human or humanized antibody sequence. The in vitro durability assay include non-specific binding assays such as BVP and DNA ELISA, AC-SINs, and plasma stability. It is suggested to be conducted and the assays before proceeding with in vivo experiments. The manufacturability assessment include integrity and stability assessment and forced degradation, which takes about 6 to 8 weeks and is suggested to be conducted for identifying PCC molecules before initiating CMC work. The purpose of in silico durability assessment is to avoid the potential durability issues at early stage. Immunogenicity, PTM, and aggregation are 3 key factors we consider the most in silico analysis. High immunogenicity can lead to ADA neutralization, hypersensitivity reaction, and cytokine storm. We can predict the T and the B cell epitope for various candidates using our in silico tools. Although the immunogenicity is extremely complex and the predict results are only useful as a reference, we may be able to avoid some high-risk sequence at early stage. Post-translational modification can affect the binding activity to the target antigen, reduce the stability of antibodies, and cause immunogenicity issue. We can analyze the common PTM sites and provide risk assessment by MOE software. If necessary, we can also conduct PTM removal, aggregation, and can cause bad performance in solubility, titer, yield, and safety features. We can predict hydrophobic region by MOE design region. We can also optimize the hydrophobic area of antibody to eliminate aggregation if needed. For the immunogenicity analysis, we have capability on both T cell and B cell epitopes. Various database and analyzing tools can be utilized to generate combined scores and results. Regarding the PTM, we will first analyze the sequence to identify the high-risk modification, such as integrin binding site, persisting, and isomerization. For the high-risk modification, we will suggest to do the PTM removal to generate the PTM removed antibodies for further validation. Here is one case study for PTM removal. The original antibody contains an isomerization site on the CER2 of the heavy chain variable region. 2 mutants were designed and tested, and the FACS results showed the S2A mutation significantly impaired the affinity and the expression level, while the D2E mutation retained these features. For aggregation analysis, we used MOE software to identify the sequence sites that are prone to aggregation. Site-direct mutations will be introduced to elevate the pro-aggregation tendencies and retain the affinity. Here is one case study of aggregation optimization. The Y-type antibody has severe aggregation. A hydrophobic patch was observed in the in silico modeling and analysis. 3 mutants are designed and expressed for testing. In the end, these mutations significantly improved the aggregation, leading to a better yield without any compromise on affinity. Besides in silico developability services, ProBio can also provide in vitro developability assessment. Some antibody drugs that have progressed into CMC or clinical stage may encounter problems with non-Swiss binding or self-interaction, resulting in side effects such as immunogenicity and reduced drug efficacy, leading to substantial economy losses. In vitro developability assays are suggested to conduct before costly in vivo animal studies to reduce these developability risks. The table below summarizes the major assay types, applications, and timelines for different assays. Manufacturability is another important assessment to select the final PCC molecule for CMC. Various stress conditions such as high temperature, low pH, and free source are suggested to be tested, and some important quality attributes such as T-melting, T-aggregation, and purity will be assessed during the processes. The case study here compared 3 candidates, and finally candidate C stands out with no major defects in the manufacturability. As a summary, Gen3 ProBio provides comprehensive antibody engineering services based on extensive bioinformatic tools of durability and functionality. We provide a one-stop durability solution, such as PDM, immunogenicity and aggregation, from prediction to optimization. Until now, there are more than 610 antibody optimization projects has been successfully delivered. Thank you all for your time, and feel free to contact us or leave a message if you have any questions.